So we are going to discuss first about steps involved in plant tissue culture. So one may say that there may be less or more steps, but I have mentioned here 6 steps. Maybe somebody will think that there we need to follow more steps, or somebody will say that we can reduce the steps. But see, basically these things we need to do during the plant tissue culture. The first step is the establish and maintenance of aseptic lab. If you don't have laboratory, you need to set up laboratory. So that will be the first thing. Or if it is the laboratory is there, you need to arrange or rearrange the laboratory as per one's need. So that is the very important thing. And then maintain the aseptic condition will be of prime importance, because under aseptic condition only you will be able to grow your plant material. Otherwise it will be competed by the what we say the contaminants or the different type of the microorganisms which may be present in that particular environment, inside bottle or inside the laboratory. Then second step is the selection of the what we say the plant material for your tissue culture purpose. There you are going to prepare that plant material. After that you are going to prepare that material. So one may say that this step may be divided into the selection of the plant material and preparation of the plant material for or explant for the inoculation purpose. Then third step is the establishment of the culture. What we say the exonic viable culture. So the culture which is free from microorganisms. So we need to take that explant, we need to prepare that explant, and in the third step we are going to inoculate on the media and we are going to allow it to grow. So when we are going to grow that what we say our explant, inoculation, and then there should be no growth of other what we say the organisms. That means no contamination must be there. So this thing is very important. And then the fourth stage where we are going to multiply, means cells will divide, or that whatever our aim is there, depending upon that there will be multiplication. In micropropagation there will be many more different plants will be formed. So that thing is going to be takes place over there. So that is the multiplication stage. And then the next stage will be the either rooting or either subculturing, depending upon your condition. So in the stage 4 you can include subculturing, or you can add it after that, and then the rooting stage will be different. So in the fifth stage here I am telling you about the rooting and development of the plantlets, again under the in vitro conditions, remember. So plant is growing now, whatever the if you talk about the micropropagation, you have developed shoot, so it will require to develop root. So we need to induce the root formation. So we need to change the media, means we need to add different phytohormone for the growth purpose. So we need to add more auxin for the root formation. So that will be that step. And then next step or the last step as per my statement will be the establishment of plants under in vivo conditions. So plants are growing under in vitro condition, under laboratory condition, and because of that what will happen, whatever our plants are there, so they are growing under ideal condition. If you will take them directly under the in vivo condition, means natural condition, they may die. So we need to take certain precautions. So all these things we are going to discuss about all these steps one by one. So step 1 is establishment and maintenance of aseptic lab. So there are several areas are there. What we say the or the work which is associated with this step, you will find that many more areas are there, and we need to maintain the aseptic conditions over there. So organization, sterilization, maintenance of PTC lab is very important. So first you need to organize the things. We have discussed about the plant tissue culture lab setup in the previous lecture, if you remember. So within that I have mentioned about this. So as per your need you can arrange the things. So whatever the what we say our laboratory setup is there, so general laboratory is there, within that you can arrange the washing area, you should arrange the where to store the chemicals, where to store the glasswares, where to prepare media, right? So all these things you are going to do. If there will be separate area, that would be good. So for the preparation of the media and for the sterilization of the media. So media sterilization will be also very important thing. So in addition to that, you may do the what we say the fumigation of all the 3 labs. So there are 3 labs as I have told you previously in the previous lecture. So general lab, inoculation room, or what we say the aseptic transport area, and the culture room. So all these rooms must be free from, at least these last two rooms, aseptic manipulation area or inoculation room and culture room, must be free from any kind of the microorganisms. So as per that need, we need to take care. If you will carry any kind of the microorganism, or if you will carry, or if your cultures will be get contaminated with the different type of the microorganisms, your culture will be destroyed. So you need to take precaution. So all these things will be very important. Then media, what we say the preparation, as I have told you in the last lecture, so preparation of the media is also an important step, and what we say the sterilization of the media will be very important. So that can be done by means of the autoclaving. So if you will autoclave it, so if you will take proper precaution, if there are certain compounds which you need to add into the media after sterilization, it is also possible, because there is another way of the sterilization of the certain components which are heat labile or which are heat sensitive. Such compounds can be filtered through membrane which is having pore size less than what we say the 0.44 microns or 0.22 microns. So which is suitable, you can use, and by means of the filtration, what we say the you will be able to sterilize your media, because the pore size of these filters is very small. So through that, microorganisms, their spores or their fragments will not be able to, or their cells will be not able to pass. So you need to do this. So these are the things associated with the step 1. So here, you will be able to see how we are going to prepare media. So see, there are different bottles in which already we have prepared the, what we say the stock solutions of our media. So you need to take them, you need to mix them in a proper amount, in an appropriate proportion, and then you need to make the final volume with distilled water. So in between you need to adjust the pH, and then you need to add the gelling agent if necessary, and then you are going to pour it into the bottle. Unlike the microorganisms culture, plant tissue culture, remember that you are going to pour the medium and then you are going to sterilize, right? So after pouring, you are going for the sterilization. So in the next step, the second step, what we say, we are going to do the selection of the and preparation of the explant. This step is also very important, because there are several works which are associated with this step, are what we say the maintenance of the aseptic conditions. So it may be possible to do this selection of the plant material, means you can grow your plant material under controlled conditions, so that there will be less chance of the contamination. Or otherwise, if you need to bring your plant material from outside, from the nature, if you are going to bring it, so what will happen? So the plant material may carry different type of the microorganisms, and that may cause contamination, that may deteriorate your culture. So you need to take care for that. So you can use different type of the sterilants. Through using that, it is possible to make it free from what we say the microorganisms. So it will be sterile. So you can use sodium hypochlorite, you can use mercury chloride, you can use hydrogen peroxide, that is H2O2, you can use what we say the ethanol, 70% ethanol, such a type of the chemicals, or many other chemicals are there also available. You can use that for the what we say the sterilization of or surface sterilization of your explant. So first select your explant, then you go for the sterilization. So remember that the concentration and time required for the sterilization may vary. So that you need to remember. In this step, so here what we are going to do is that establishment of exonic or viable culture. So work associated with this step is that we are going to develop or establish our culture. So we need here at this point what we say the laminar airflow, because we need to inoculate our what we say the explant onto the media. So to do so, we need to whatever we have sterilized, surface sterilized our explant, that we need to inoculate. So to do that, we need certain type of the cabinet known as what we say the laminar airflow, and within this laminar airflow, so aseptic conditions are maintained, or aseptic environment is maintained. How it is maintained? So I have told you already about that in the last lecture. So the by means of the suction, air is suctioned and that is passed through the filter, what we say the HEPA filters, which are having very small pore size, and because of that filtered air will be there in that cabinet which is coming, and because of that there will be no what we say the chance of the contamination, and within that area we are going to inoculate our explant onto the media. So these precautions we need to take over there. So this is the thing, what we say, we can see people are doing the inoculation work over there in the within the what we say the laminar airflow. So they are using laminar airflow, they are having separate laminar airflows. So they may be vertical or horizontal type of the laminar airflows may be there, remember. So these laminar airflows are used for the inoculation purpose. So next step is the multiplication or subculturing stage. What you are going to do, because now you have inoculated your explant on the media, so it will respond to that what we say the media, and first it will get acclimatized to that particular environment, remember, because this what we say the in vitro condition is new for that plant material, and because of that or that cells, and that within that condition it will take some time to what we say the grow. So first it will prepare itself for the cell division or for growth, and then we need to, after establishment, what we are going to do, we are going to transfer it to the another media which is known as the multiplication media. So in this multiplication media we are going to maintain, if for example if you want to make shoots, you will maintain more amount of the cytokinin, and because of that the plant cells will grow and that will develop into the shoot. So this thing is going to happen within this stage. So once you are going to, for example, if you have inoculated few cells or tissue or small part of the plant, so it will produce several shoots. So several shoots you can take to another bottles where again you are going to multiply it. So because of that it is known as a multiplication stage, and here you need to maintain the aseptic conditions. Again you are going to do all this work under the what we say the sterile environment, that is inside the laminar airflow. So here you are able to see the similar type of the picture, the same type of the work. Subculturing is done over here. Next step is the stage for rooting and development of the plantlets. Now you have developed the shoots, they may require the formation of the root. So you need to what we say add another media, or you need to inoculate your cultures or your plantlets to the another media which will proliferate the roots. So root initiation can be done using the oxygen. So you need to increase the concentration of the oxygen, and through that the roots will be formed. So you need to subculture again, or you need to transfer your plant material to the another bottles where there will be root induction. So that will happen within this. So work associated with this step will be the maintenance of the again sterile condition. If there will be any bottle is contaminated by chance or by mistake, so you need to remove it, you need to autoclave it, you need to destroy that, remember. So that is also very important thing. Last stage where you are going to establish your plantlets under in vivo condition, that is natural conditions. So you need to take all these plants to the what we say the polyhouse, where you are going to maintain initially very high amount of the humidity and low amount of the light, remember, because plants are grown under ideal condition, and because of that they may be not able to survive in that new situation, new condition. So you need to maintain high humidity because tomatas are open continuously, and because of that they will lose the water, and because of that it may result into the wilting and the death of the ultimately of that plant, and because of that so we need to maintain certain type of the conditions. So possibly, so here also you may go for the maintenance of the conditions, so that there will be no what we say the pathogenic attack of the different type of the bacteria, or what we say the viruses, or the different type of the fungal pathogens who should not attack, and because of that you according to that you should take care. You should use certain kind of the antibiotics or certain type of the chemicals to control it, right? So entry here also is restricted. Too many peoples are visiting this area is also not good. So you need to take care about that. Okay, so regular workers, so they know about that what kind of the what we say the precautions they should take, so they are going to follow. Because of that, so such a type of the peoples are only allowed. So likewise, so these are the 6 different steps of what we say involved in the plant tissue culture. So ultimately now plant is growing outside in the nature, right? So now the plant is ready to transfer to the field. So here you will be able to see such a type of the hardening or acclimatization facility within the polyhouse. There within the polyhouse there are small domes are prepared, where so that why these small domes are prepared, remember, so that there it is possible to maintain a high humidity. So humidifiers are there, you are able to see in this picture also humidifiers are there, but these humidifiers, what we say, so large area will be you need to maintain under the high humidity condition, and to avoid that, so small domes are prepared. In general you will find it at the hardening facility, this type of the small domes are prepared, so that within that small area you will be able to maintain the high humidity. So this is about the all the steps of the what we say the which are involved in the plant tissue culture. So this is a very important to understand these different steps which are involved in the plant tissue culture.